Physiology Smart Art video, where we guide you through an important piece of art. After watching this video, you should be able to summarize the process of transcription. Transcription is the process of producing RNA from a DNA template. The term transcription is appropriate because transcribe means to copy or rewrite. Genetic information is encoded in a length of DNA called a gene, which you can see here. The process of transcription rewrites this genetic information into the molecular language of RNA, which is represented here. There are many types of RNA, but here we will focus on the transcription of messenger RNA, or mRNA, which carries the information, the message, needed to synthesize proteins. Let's start by looking at the first step in transcription, gene activation. Gene activation begins once the DNA strands have separated, and the enzyme RNA polymerase binds to the control segment, or promoter, on the first segment of a gene. The strand of DNA used to synthesize the RNA is called the template strand. In the second step, we can see RNA polymerase promoting hydrogen bonding between the nitrogenous bases of the DNA template strand and the complementary RNA nucleotides. The RNA nucleotides are then linked by covalent bonding. Be sure to notice how the DNA and RNA nucleotides are aligning as complementary base pairs. But remember, RNA contains a nucleotide uracil, which binds with adenine. So we see A with T, U with A, and G with C. A group of 3 mRNA nucleotides is called a codon, and each codon codes for a particular amino acid in a protein. Translation ends in the third step, when the DNA template strand reaches a stop signal. The newly formed mRNA strand detaches, as can be seen here, and the DNA strands reassociate as hydrogen bonding occurs between the complementary base pairs. The resulting mRNA strand, however, is still considered immature, so it is in a final process that the mRNA is edited before it leaves the nucleus to direct protein synthesis. Represented here are lengths of mRNA that are non-coding. These are called introns and are snipped out. The remaining coding segments, or exons, are spliced together to create a shorter functional strand of mRNA. To summarize, in transcription, a process that occurs entirely in the nucleus, a length of DNA called a gene is rewritten into the molecular language of RNA, and in this case, messenger RNA. The mature mRNA exits the nucleus and enters the cytoplasm to begin translation, the next step in protein synthesis. We will discuss translation in another Smart Art video. You may ask, so what? Why is it so important for DNA to be transcribed properly into mRNA? The answer is that mutations, small permanent changes in the nucleotide sequence of a gene, can have major impacts on health. Serious disease, such as sickle cell anemia, can result. In this inherited disease, the gene codes incorrectly for a single nucleotide. As a result, the protein hemoglobin is not properly constructed, and the red blood cells are not fully functional. Patients suffer from a host of symptoms, such as breathlessness, severe pain, organ damage, and even premature death. Another Anatomy and Physiology Smart Art video, where we guide you through an important piece of art. After watching this video, you should be able to summarize the process of translation in the synthesis of proteins. Recall that translation is the final event in protein synthesis after gene activation and transcription. It's also important for you to remember that the first 2 steps, gene activation and transcription, occur here in the nucleus, while this final process, translation, occurs here in the cytoplasm. The term translation is appropriate, as this step is all about translating genetic information from messenger RNA, or mRNA, to the language of proteins, which is a sequence of amino acids in a polypeptide chain. That is represented here as a series of 3 balls, each of which represents an amino acid. Here you can see a small segment of the mRNA strand that exited here from the nucleus. This codes for our end product, a polypeptide chain. Let's start with the language of nucleic acids, a sequence of nitrogenous bases, by taking a closer look at the initiation process. Here you can see the nitrogenous bases grouped into sets of 3, and each set makes up what we call an mRNA codon. Each mRNA codon, once it is translated, designates or codes for a specific amino acid that will be inserted into the growing polypeptide chain. All of this is shown nicely in this particular image, as you can see the mRNA and representative codons and how that relates to a particular amino acid. Again, it's important to remember translation occurs outside of the nucleus, in the cell cytoplasm. Recall, the nucleus contains DNA and the newly synthesized mRNA. This mRNA then exits the nucleus through a nuclear pore, which is represented here in the image. Let's go back to the cytoplasm and now talk about the ribosomes, one of which is represented here. Notice that inside each ribosome is a series of 3 tRNA binding sites, named E, P, and A, on what is called the large ribosomal subunit. That can be seen here. See here how the mRNA strand binds to the P site on the large ribosomal subunit. Let's now see how transfer RNA, or tRNA, shuttles amino acids to the ribosome and binds its anticodon with the mRNA codon in a complementary fashion, U with A with U, and C with G. The small and large ribosomal subunits interlock around the mRNA strand, forming what we now call a functional ribosome. The initiation complex is now complete and protein synthesis proceeds. Elongation occurs when the next tRNA molecule arrives at the A site, here, again binding anticodon to codon. The adjacent amino acids form peptide bonds, the ribosome moves farther along the mRNA strand, and the first tRNA moves from the P site to the E site and is released into the cytoplasm. Elongation continues as perhaps hundreds of amino acids are sequenced, but ends when the ribosome reaches the stop codon at the end of the mRNA strand. Termination occurs as a protein releasing factor, not shown here, recognizes the stop codon, and the polypeptide is released. This newly synthesized polypeptide is then processed into complex shapes, resulting in a functional protein, such as hemoglobin or perhaps collagen. Remember, proteins are the most abundant organic components of the human body, and in many ways, the most important.